Let's get started. It's my pleasure today to introduce, well, I think you all know him already, Arek Kulczyk, one of our intramural faculty members at IQB. Just a very brief, some points of his career. Master's at Jagiellonian University in Krakow, went from there to the MRC LMB, that storied institution where so many of the luminaries from our field have passed through, and then spent over a decade at Harvard, rising through the ranks, postdoctoral fellow, and then an instructor. So from a Harvard faculty to Rutgers faculty, and he moved his laboratory here to Rutgers. You may have seen his work in top journals like JBC and PNAS, a lot of great work on the replisome, which you'll hear about today, so I won't spoil any of that. But I have to say that working here in Cryo-EM in Rutgers, I'm getting a lot of opportunity to interact with Arek and see what he's up to. And I think we can all be excited about some really exciting new methods, new technologies, and most of all, new biology that he's bringing to the university, and new directions that he's moving into. So can't wait for this talk, and then lots of great work to come.
Well, good afternoon, ladies and gentlemen. Jason, thank you very much for a very kind introduction. And thank you, organizers, for giving me this opportunity to present our data. Today, I will present the structure of the replisome. The replisome is a highly dynamic, multi-protein molecular machine with components constantly changing. This intrinsic dynamics and complexity pose a number of challenges for structural studies of the replisome. As the consequence, the replisome has been resistant to structure determination for a very long time. It has changed recently due to the breakthroughs in the field of cryo-electron microscopy that allowed us to determine the structure of the phage T7 replisome. There's so many screens here. Bacteriophage T7 provides a very good model system to study replication, and its replisome is a good target for structural studies because it's relatively simple. Only 4 proteins are required to form a basic phage replisome, yet the reconstituted complex recapitulates all key features of more complex prokaryotic and eukaryotic replisomes. So this slide summarizes our current understanding of the roles which these proteins play at the replisome. On DNA leading strand, gene 5 DNA polymerase, shown here in green, is responsible for actual polymerization of nucleotides. E. coli thyrodoxine, shown here in yellow, functions as a processivity factor for DNA polymerase. The structure of the DNA polymerase thyrodoxine primer template complex have been determined by X-ray crystallography and shown below. DNA polymerase thyrodoxine holoenzyme cannot, however, polymerase through duplex DNA, hence the requirement for the helicase activity of gene 4 helicase primase, shown here in blue and red. DNA helicase assembles on the DNA lagging strand and translocates from 5' to 3' direction using energy derived from hydrolysis of deoxythymidine triphosphate. In addition to being the helicase, gene 4 protein is also the primase, and as such, it accounts for initiation of the lagging strand synthesis. At specific pentanucleotide sequences on the lagging strand, the primase domain catalyzes synthesis of short RNA primers, shown here in green. These primers are then extended to a Okazaki fragment by the lagging strand DNA polymerase, shown in yellow. Gene 2.5 protein is a single strand binding protein binding to the single strand elements within the lagging strand. This protein is also important for coordination of the DNA synthesis. The reverse polarity of 2 strands in the duplex DNA pose a topological problem for their simultaneous synthesis, because polymerases can extend the DNA only one direction. So in this particular case, the leading strand enzyme would like to move from right to the left, and the lagging strand enzyme from left to the right. So how do we solve this puzzle? In 1980s, Bruce Alberts proposed a model for the replication fork in which all proteins come together to form a single complex. In this model, actually, I just forgot to show you all the beautiful crystal structures of helicase in single strand binding protein. Let me do this, and then I go back to the Alberts model. So the structure of the helicase has been determined in two oligomeric states, as the hexamer and as the heptamer. The heptamer is an active form, whereas hexamer, as I mentioned, binds to single-stranded DNA. Now, the structure of the gene 2.5 protein is also available and shown below. This protein is missing from our Cryo-EM structure. So now, I apologize for that. Let me go back to Albert's model. So in this model, the lagging strand DNA polymerase is brought to the replisome by having the lagging strand to form the loop. Now the replisome can move in one direction while synthesizing both strands. I started working on this project by identifying conditions required to form the replication complex. So what we needed was a DNA ligand resembling the replication fork, where the leading strand and lagging strand proteins could bind. I obtained such a DNA, but by folding a single-stranded template such that the stable tetraloop is formed in the middle of the sequence, flanked by the short alpha helix. Two overhangs are also formed, and after annealing DNA primers to these overhangs, they represent the leading and the lagging strands. Both primers contain dideoxynucleotides at the 3' ends. In the presence of the next incoming nucleotide specified by the template, so in both cases, there will be deoxythymidine triphosphate, DNA polymerases bind tightly to these primer templates. And we expected that primase helicase will bind to a single-stranded element located within the lagging strand. I assessed folding properties of this DNA ligand by using fluorescence melting spectroscopy in the presence of the fluorescent dye, which intercalates into double helix. And the results are shown below. The decrease in the fluorescence signal as the function of the increasing temperature suggests melting of the duplex DNA and release of the dye to solution. This biphasic melting profiles across the range of DNA concentrations indicated by different colors of curves is indicative of formation of single monomeric species in solution with the melting temperature of 53 degrees, as it can be calculated from the maximal first derivatives of the melting curves. In contrast, the DNA ligand, lacking this stable tetraloop sequence, does not display this biphasic melting transition. All proteins in this complex are genetically altered. DNA primase helicase contains a single amino acid substitution in its nucleotide binding pocket located at the interface between neighboring subunits within the ring. Glutamic acid 3,4,3 functions as a catalytic base for hydrolysis of the incoming nucleotide. The ATP analog was used to trap the structure. Because it's in position to activate the water molecule, which is not visible in the structure, for the nucleophilic attack of the gamma phosphate of the incoming nucleotide. So the reversal of charges at this position from the glutamic acid to glutamine renders the protein inactive. The protein can bind nucleotides, however, cannot hydrolyze them, and subsequently cannot translocate along single-stranded DNA. DNA polymerase is also genetically altered. I'm not showing on the slide. It contains two amino acid substitutions in its exonuclease active site. As the result of these mutations, exonuclease activity is reduced by the factor of 10 to the 6th, whereas the polymerization activity remains the same. Upon additions of replication proteins to fork DNA, a stable complex is formed, as evidenced by the buildup in anisotropy signal of a fluorescently L-bit template. The mutant primase helicase binds to DNA with the affinity 4 times higher than the wild-type protein. I investigated replisome samples by negative stain EM. First, I wanted to confirm the presence of DNA polymerases in the complex, and so I labeled thyrodoxine DNA polymerase holoenzyme with gold particles. Negative stain reveals the presence of 1 or 2 gold particles colocalizing with the complex, confirming the presence of DNA polymerase there. Now I have to emphasize that this labeling with gold is not quantitative. Likewise, I wanted to confirm the presence of DNA in the complex, and for that reason, I developed this novel labeling scheme of DNA with quantum dots using a streptavidin biotin linker. The micrograph on the left shows quantum dot DNA conjugates stained with uranyl formate. After addition of the replication proteins to these quantum dot DNA conjugates, we observe replisomes colocalizing with quantum dots. It's probably better visible on these images of single particles picked from these images. So for instance, if you pay attention to the zoom in image, you will see the helical ring with two densities corresponding to polymerases and the quantum dot colocalizing the center of the ring. With the assembled replisome, we acquired approximately 10,000 images using a Titan Cryo instrument shown here. I would like to mention this instrument, this particular instrument was not equipped with a direct detector. Nearly 750,000 particles were then automatically picked from these images as shown here. The picked particles are indicated by red circles. And the structure was calculated in the program RelyOn, which uses Bayesian statistics. Such approach is very computationally expensive. And so I was actually very fortunate that I obtained 3 grants from NSF and from the program called Exceed that gave me access to Stampede supercomputer in Texas. This picture shows some of the computing racks from Stampede. Just let me give you a couple of examples illustrating the scope of these calculations. I started this calculation using a 64 CPU Linux cluster. I was hoping that there would be sufficient for structure determination, but it turned out I was completely wrong, and this was one of the first wake-up calls. So in the first step of image processing, the particles need to be picked up from the images. So I submitted this job to the cluster, and this is a time estimate which I got in return. So what it says that I have to wait over 7,000 hours for the job to be completed. So I was very tempted to do this, but that actually corresponds to more than 10 months. So it's not really possible. So we solved this problem eventually by submitting thousands of small jobs instead of one big job to the cluster. But for that, we needed a supercomputer. Next step in the image processing is a classification of peak particles into a number of groups shown here. These groups represent 2D projections from the 3D structure. To complete one round of such calculation with 750,000 particles, I needed to use more than 4,000 CPUs for about 2 days. So it turns out that in this version of RelyOn, high memory demands are a bottleneck with approximately 60 gigabytes of memory required per one core. Now this problem has been circumvented to the large extent by rewriting the program into the GPU. So we're all now using GPUs instead of CPUs. Structure calculations in RelyOn require a starting model. And since the majority of these 2D classes reveal the presence of the hexameric ring of DNA helicase, I used atomic coordinates of the hexameric helicase to generate a starting model. These coordinates were filtered to 60 angstrom resolution. With this starting model, I was able to calculate the structure. This slide shows an electron density map. This electron density map can be fitted with atomic coordinates of hexameric helicase, shown here. The structure also reveals the presence of 2 polymerase molecules, one on the top, the other on the bottom, asymmetrically binding to helicase. Likewise, associated primase domains bind to the helicase in an asymmetric manner. So now let me move on and show you more details. So this slide shows the 3D map on the left, then fitted atomic coordinates to this map in the middle, and surface representation on the right. DNA helicase is shown here in blue. DNA polymerases are in green. Each DNA polymerase contains exonuclease domain, and it's in purple. Primases are orange, and processivity factor thyrodoxine is shown in yellow. The structure reveals that 2 molecules of DNA polymerase bind to the helicase at the back of the replisome relative to the direction of unwinding by DNA helicase. So in this case, the repisome would move from the left to the right. I'm sure you have already noticed that the structure does not reveal a clear density representing the DNA. However, the conformation of this replication protein strongly suggests its presence. So I already discussed oligomeric properties of DNA helicase. Hexamers are observed in the presence of DNA, and I will get back to the conformation of DNA polymerase later on during my talk. In addition, the structure reveals the density, which is unassigned and located in the middle of the hexameric ring. Previous biochemical studies showed that DNA binds in this channel. So the obvious question is, why don't we see the DNA? My best guess is there is still some heterogeneity in this sample. So one can envision a scenario in which the DNA is bound by different subsets of subunits from the hexameric ring of DNA helicase. So by averaging thousands of particles, and the DNA is bound a different way to each one of them, that would basically lead to canceling of the density of the DNA. So obviously I was interested in how the replisome samples would look like in the absence of the DNA. And so I performed negative stain experiments. In the absence of the DNA, DNA helicase is present in 3 different oligomeric forms, as hexamer, heptamer, and octamer. These higher oligomeric forms, heptamers and octamers, were not observed in the replisome samples. In addition, we couldn't detect the binding between DNA polymerase and DNA helicase, again, suggesting that the DNA is present in the complex. So basically, my belief is that this complex cannot be formed in the absence of the DNA, and it's actually supported by the data. So the resolution of this structure is 13.8 angstroms, and I calculated it using a so-called gold standard approach from the Fourier shell correlation curve according to 0.143 criterion. So this information is for some of you Cryo-EM lovers. So this is relatively modest resolution at this time. However, please remember that this is a new structure. It is asymmetric. It is highly heterogeneous. I actually had to assemble on the EM grid. And the data were collected with the microscope, which was not equipped with the direct detector. So since then, I actually recollected this data using our beautiful microscope located here in the IQB. This is a 200 kV instrument equipped with a K2 direct detector. So I'm currently analyzing this data. I'm very hopeful that the resolution will be increased and then we'll visualize the DNA. I won't be presenting this data today. This picture on the left side of the slide shows that the reprojection from the 3D structure in these black rows are in good agreement with experimental class averages, which is the way to support the notion the structure is actually correct. I would like to also add one more information here, that the handedness of the map was determined by using random canonical King experiment, but also there's information for some of you who are interested in Cryo-EM in more details. Well-defined electron densities allow for fitting of 15 sets of atomic coordinates representing replication proteins into the map. This slide shows the local cross-correlation coefficients calculated between the map and these PDBs and mapped onto the surface of the replisome. With the red color representing bad fit and blue color representing good fit. So as you can see, the overall fit is pretty good. The first row shows the map of the replisome, and the bottom row shows this PDBs fitted into it. Now, I'm sure that you are aware that fitting atomic coordinates automatically to maps in this resolution is actually not trivial. It's actually one of the challenges the field facing now. It's sort of a great zone for Cryo-EM. We don't really have very good methods to doing this in a really well-defined systematic way yet. The structure reveals asymmetric binding of the leading and the lagging strand polymerase to the ring of primase helicase. The leading strand DNA polymerase binds to the replisome through the helicase domain, as shown here in this insert, whereas the lagging strand polymerase binds to the replisome mainly through primases. Now, one question which you might be asking right now is probably, how can I distinguish between leading and lagging strand polymerases based on the structure alone? And the simple answer is that I cannot. So this distinction was made based on the additional evidence. I determined low resolution structures of individual leading and lagging strand complexes, and also perform a number of biochemical experiments which support this assignment. I can get back to this at the end of the talk if you are interested. With the assignment of polymerases, we can propose the model for the replication for it. DNA helicase unwinds a duplex DNA, separating into two strands. The resultant lagging strand passes through the central channel in the helical ring. It's better visible here on this insert. The DNA has right polarity orientation for binding by one of the primase domains, so it can synthesize the primer, and then by binding by the DNA binding crevice of the lagging strand polymerase. The leading strand is displaced by negatively charged surface of DNA helicase, so it can be bound by the leading strand DNA polymerase shown here. This model is supported by extensive mutational analysis. I'll just show you one of these mutations we made just for simplicity. The phenylalanine 523, which contacts the extruded DNA strand, the mutations of this residue abolishes strand displacement synthesis of the complex. Also, all regions involved in DNA binding are positively charged in otherwise negatively charged replisome. So now the question is, what does the structure actually tell us? The geometry of binding between the replication proteins suggests the mechanism for formations of the replication loop. As I mentioned, the lagging strand in a polymerase, so that would correspond to this part of this complex, binds to the replisome through one of the primases. And this interaction positions the lagging strand polymerase such that the polymerization activity of this enzyme would push the newly synthesized strand towards the central channel of the DNA helicase. Consequently, this activity combined with the movement of the single-strand DNA through the central channel account for formations of the replication loop. So actually, when I analyzed this complex and realized that, I was quite amazed. I mean, this is really beautiful and simplistic solution. And a lot of groups just imply a lot of conformational changes just leading to formation of the replication loop. I see it now as a natural consequence of the geometry of binding between these proteins. Enzymatic events on the leading and lagging strand are not equivalent. The lagging strand polymerase repetitively dissociates from DNA and reassociates with the newly synthesized primer upon completion of each Okazaki fragment. In addition, the primer synthesis is a very slow enzymatic event. We actually measured this in the single molecule experiments. And at the time required for formations of the primer, approximately 1,000 of nucleotides would be synthesized on the leading strand. So that raises immediate question why the leading strand synthesis does not outpace the lagging strand synthesis. Well, two models have been proposed. In the first model, the entire replisome transiently stops. So it gives time, the lagging strand DNA polymerase, for the completion of the slow enzymatic events on the lagging strand. In the second model, both DNA polymerases synthesize the DNA at different rates. Believe it or not, actually, both models have been supported by experiments, single molecule experiments, but in different systems. Obviously, the structure, this is a single snapshot from this process, does not allow us to exclude between these two models. However, the geometry of binding between the proteins actually supports and explains the second model. So in this second model, the leading strand polymerase would, the rate of synthesis displayed by the leading strand polymerase would depend on the rate of unwinding by DNA helicase. However, the lagging strand polymerase and its rate of synthesis would depend on the amount of single-stranded DNA available in the replication loop. This model, actually, the second model, has been proposed by Smita Patel, who is one of the faculty at Rutgers. So next question is, how are these loops released? Again, two models have been proposed. In a so-called collision model, the collision of the lagging strand DNA polymerase with the 5' end of the preceding Okazaki fragment triggers loop release, bang, just like that. In the other model, so-called signaling model, primer synthesis is the event triggering loop release. Now, the nature of this signal is actually unclear, and our structure provides some clues. As I mentioned, the lagging strand DNA polymerase binds to the replisome through the contacts with one of the primase domains, shown here. We speculate that disruption of this interaction triggers loop release. We could envision the following scenario. DNA, the primase dissociates from polymerase and binds to the primer recognition site, which was just exposed by the DNA helicase. This simple switch mechanism could be mediated by different binding affinities of primase to DNA polymerase and primase to DNA. That, in turn, would lead to the release of DNA polymerase from the replisome and release of the replication loop. So these are only models. So we actually are trying to verify them experimentally. Now, what structure reveals is that the leading and the lagging strand DNA polymerases contact one another. I would like to remind you that the overall fold of DNA polymerase resembles the right hand. So if you pay attention on this picture for a moment, you will see that the polymerase has fingers, palm, and thumb. So this direct protein-protein interaction is mediated through the palm domain of the leading strand polymerase and through the fingers of the lagging strand polymerase. And this interaction can provide the mechanism for coordination of DNA synthesis because changes on one strand can be directly transmitted to the other. How would it work? Well, interestingly, the leading strand polymerase reveals the closed conformation in which fingers are located in close proximity to the thumb domain. Such conformation has been observed upon binding to a primer template. In contrast, the lagging strand DNA polymerase shows the open conformation in which thumb rotates outwards from the fingers. These conformational changes in the polymerase structure would provide the sensing mechanism detecting binding on dissociation on DNA on one strand and the other, hence could provide the mechanism for coordination of synthesis. Now, one more point which I would like to make here is that the geometry of binding between the lagging and the lagging strand polymerases exclude the possibility of another molecule of DNA polymerase binding at the fork, explaining perhaps why only two polymerases can be actively involved in the synthesis of the DNA. It has been somehow somewhat controversial question in the field how many polymerases actually bind to the replisome. My take on this is that more polymerases can bind to replisome, but only 2 of them can actively synthesize the DNA. So with that, I would like to acknowledge people who contribute to this talk. First, I would like to thank Charles Richardson, my former boss from Harvard Medical School, for his support and making things possible. Piotr gave me access to SB-Grid, where I carried out structure calculation at the initial stage of the project. Peter Meyer helped me with these calculations. Arnemele helped me to collect data at Aarhus University in Denmark. And Bridget Karager and Clint Potter gave me access to the microscope at Scripps. And I used this data to collect low resolution structures, which I haven't shown today. And if you allow me, I would like to advertise my lab a little bit, which is just opening and we are hiring. And the next goal is to determine the structure of the human mitochondrial replisome. So if you are interested, just please follow up with me after that. Thank you very much for your attention, and I'll be happy to take questions if you have any.
So I'm Vlad Kolodich from Office of Dissociation of DNA. So Arek, it's a very impressive work and beautiful with graphics and.
Thank you, sir.
Yeah, thank you. Yeah. So I have a question, more technical question for you. So you mentioned, because I'm from Office of Advanced Computing, so we have a supercomputer here as well. So that is MRL, and we maintain over 10,000 cores. So we are kind of like on par with Stampede. My question is, are you aware of it? And did you try to run your jobs on MRL? Because now we have Reliant installed as well, which is, I believe, the latest version 2.1.
Yes, that's correct.
And that is GPU version, right?
Well,
so
my first reaction would be, this is totally awesome. So thank you for installing Rely on this. So answering first question, yes, I'm aware of the supercomputer. And question 2, I would love to actually use it. I haven't used it yet. I just simply didn't have time to do that yet. But I would love to follow up with you, actually.
Sure, yeah,
do you
definitely.
have some GPU nodes?
Yes, we have GPU nodes, so you can run it on GPU cards as well.
I'll be really interested in actually following up on this conversation. Thank you very much for bringing this up.
Thank you for your report. I have a question about the DNA in your structure. It seems that you don't have DNA density. Is that at all, or you have some density for the DNA? My interesting is about there should have some DNA interact with the DNA polymerase. Do you have like double strand density, single strand density, or like don't have any of them? And do you have an explanation for that? Thank you.
Thank you for this question. Well, first of all, you sound like one of our reviewers. So basically you're saying that there is a DNA there which we cannot see, right? Yes, so this is basically what I'm saying. So, okay, I can actually go back to this. I bet this movie will drive you nuts at some point. So there is a density which is unassigned. So let me just go back there. That's right here. Right here in the center of the ring. Now, your question is referring to any density located in the proximity to the leading strand in the polymerase, which you expect to see, right? So the question is actually, I don't really see it. Okay, I don't see this density. So the DNA polymerase would adopt the close conformation in the presence of the DNA, right? In the absence of the DNA, it would be opened. DNA is what really traps the helicase in the presence of the incoming nucleotides, dideoxynucleotides in this conformation. So my take on this is actually that it is a suggestion that the DNA is present there. but we don't see it. Yeah. Again, my feeling is that there is the heterogeneity in the sample, which obviously is reflected by the resolution. And I hope to be able to sort these particles out by analyzing the data collected with direct detector. I know that I don't answer actually your question, but I think there is just no answer to your question. It's a very good one. Thank you.
Hi, Eric. That was
Hello,
nice.
hi, Kathy.
That was great. I'm curious to know, since the resolution wasn't that high, presumably there was some heterogeneity in your particles. Did you play the game of subclassification, and was it informative?
Yes, I did play this game, and the bottom line is that it just didn't really turn out very informative. So this actually course of action was also, I mean, they were reported one structure at the end was sort of suggested by reviewers. So at the beginning, I was working with 4 classes during the 3D classification, and consequently, these classes did not really contain the high number of particles. So, you know, one of the questions was whether the structures are representative for the entire data set. So actually end up including more particles in the structure which we reported, which also hit the resolution. But I think really direct detectors is the answer to this question. I mean, I think this new data set will allow me to calculate more, pull out more structures of the data set because it is heterogeneous. Okay.
You mentioned that you used an ATP analog to get the structure, and that you also mutated the ATP hydrolyzing residue. Why was it necessary to do both of those things? It seems like one or the other would be sufficient.
Oh, you are referring to DNA primase helicase mutation?
Yes.
Yeah, so I think I confused you during this presentation. I apologize for that. So this ATP analog was used by, I think, Arek and Tom Ellenberger to crystallize the complex. Okay, but in the structure of the replisome, I use actually DTTP. It wasn't an analog. So even in the presence of DTTP, primase helicase is inactive. It cannot crystallize, it cannot hydrolyze it because of this mutation from glutamic acid to glutamine. Thank you.
Hi, was a beautiful work and nice talk. So I have a question about the different speed of the polymerization in the leading strand and the lagging strand. Has anyone measured actually the replication rate by this replisome versus just polymerization rate on the single-stranded DNA by purified DNA polymerase? And actually, what you're saying is the replication rate by the replisome is slower than that the polymerase can just do polymerization on the single-stranded DNA template. Is it the case?
So my first instinct is just let's do it together. It hasn't really been done yet. But what has been done is I don't believe that the rate of polymerization displayed by polymerase only has been measured. I mean, it's not a very processive enzyme without the helicase. So we actually measure the rate of polymerization displayed by the helicase polymerase complex on the leading strand when the synthesis continues. And the average number is 100 nucleotides per second. And it's a Gaussian distribution. You can see some molecules at 30, some at 300. Average is 100 plus something. Now, for the replisome, that's also a tricky question. So, how do you know that actually your replisome does what it's supposed to do in the single molecule assay? So we developed such an assay in which we can measure actually formation of the replication loops. Okay, so when these replication loops are formed and the primers need to be synthesized, and takes, I think, 6 seconds was the number we measured. So ultimately, it's obviously slower than only leading strand complex. I think the method that you are developing will really nicely fit into this picture, because it's very difficult to isolate this individual rates from the entire complex. Yeah, accessing this question, the last question, how quickly the lagging strand polymerase goes and how quickly the leading one goes, it's not that easy. That's a good system, good model system, I think, for this sort of research.
All right, thank you, Arek, once again for your talk today. And thank you all for attending. Have some coffee on the way out, and we'll see you at the next seminar with your usual host, Sijen Wang.
Thank you.